In lecture number 4, we're going to learn about hemostasis. Now, this is the process where then we have the stoppage of bleeding resulting from a break in a blood vessel. Now, normally blood flows smoothly past the intact lining of the blood vessel walls. But if a blood vessel wall breaks, a series of reactions is set into motion to accomplish hemostasis, or stoppage of bleeding. This response, which is fast and localized, involves many substances normally present in plasma, as well as some that are released by platelets in injured tissue cells. Hemostasis, the multi-step process, begins when a blood vessel is damaged and connective tissue in the vessel wall is exposed to blood. This is pretty much a very detailed organization of how all this process occurs, but we're going to go into just a little bit more. Now, the immediate response to blood vessel injury is vasoconstriction, which causes the blood vessel to go into spasms. Now, these spasms narrow the blood vessel at that point, decreasing blood loss until clotting can occur. Now, platelets are repelled by an intact blood vessel wall, but when it is broken so that the underlying cartilage fibers are exposed, the platelets become sticky and cling to the damaged site. Anchored platelets release chemicals that enhance the vascular spasms and attract even more platelets to the site. As more and more platelets pile up, a small mass called a platelet plug forms. At the same time, the injured tissues are releasing tissue factor, a substance that plays an important role in clotting. PF3, a phospholipid that coats the surface of the platelets, interacts with TF, that's the tissue factor, vitamin K, and other blood protein clotting factors, and with calcium ions to form an activator that triggers the clotting cascade. Now, this activator is called the prothrombin activator, and it converts prothrombin, present in the plasma, to thrombin, which is an enzyme. Wow, say all that real fast. Now, thrombin joins soluble fibrogen proteins into long hair-like molecules of insoluble fibrin, which forms a meshwork that traps the red blood cells and forms the basis of the clot. Within the hour, the clot begins to retract, squeezing serum from the mass and pulling the ruptured edges of the blood vessel closer together. This shows a fibrin clot. You can see the red blood cells trapped in a mesh of fibrin threads. Normally, blood clots within 3 to 6 minutes. As a rule, once the clotting cascade is started, the triggering factors are rapidly inactivated to prevent widespread clotting. Eventually, the endothelium regenerates and the clot is broken down. Now, despite the body's safeguards against abnormal clotting, undesirable clots sometimes form in intact blood vessels, particularly in the legs. A clot that develops and persists in an unbroken blood vessel is called a thrombus. If the thrombus is large enough, it may prevent blood from flowing to the cells beyond the blockage. An example would be a heart attack if a blockage occurs in the heart. Now, if a thrombus breaks away from the vessel wall and floats freely in the bloodstream, it becomes an embolus. Now, an embolus is usually no problem, unless or until it lodges into a blood vessel too narrow for it to pass through. An example would be an embolus that travels to the brain and thus causing stroke. Thrombocytopenia results from an insufficient number of circulating platelets, even normal movements called spontaneous bleeding from small blood vessels. The term hemophilia applies to several different hereditary bleeding disorders that result from a lack of any of the factors needed for clotting. So, what are the key points in this particular lecture? First of all, what are the steps of hemostasis and explain the process? What are the types of undesirable clotting? And what are some types of bleeding disorders? This ends lecture number 4. We're going to move on to lecture number 5.